Okay, welcome again to experimental psychology lecture with Polly. Today is the agenda today is to okay, two main things uh, that are in that are part in our agenda. So one is to review the within subjects design using paired t-test, and one is to uh, review the within subjects design with demonstration on using repeated measures ANOVA. Can anyone tell me the main difference as to why we're using paired t-test and repeated measures ANOVA? It is the same logic. It is analogous to the difference between using independent samples t-test and and using one way between subjects ANOVA. How about for this one? Can anyone tell me the main difference for using such? Referring to the levels of the IV, right? So in the paired samples t-test, we only have two levels of the IV, whereas for the repeated measures ANOVA, we have two or more levels of the IV. Since we are referring to within subjects design, is correct in mentioning that the measurement of the dependent variable is measured twice in the paired samples t-test, whereas in the repeated measures of ANOVA, it's more than two times. Okay, so let's proceed to our lecture. So we now have actually if we're going to look at the different textbooks in experimental psychology, um there's no hands-on very rarely do I notice a focus on um how statistical analysis play out in the textbook. But then again, my approach is pragmatic or very practical according to the things that you need when you um if you are to propose an experiment as your final output. Of course, you need to understand well um, how to read journal articles, psych research papers using experimental method, and you you do not shy away on the methods and the results section, because those are the crucial points for you to establish your critical thinking when you try to integrate the evidences you have gathered from the past literature or reported studies. So if you shy away from that consistently, then you cannot formulate critical thinking as to why there is it will be difficult for you to formulate a sensible research problem. Okay, so in our calendar we have plenty of time for consultation on your final output. That's why um I won't be uh, uh you won't have any reasons for not giving me updates, because we have allocated ample time for your final output. Okay. So the learning objective today is to conduct analysis using repeated measures design by conducting paired t-tests. And then how do we interpret its results? And I'll provide you a sample of writing results using in APA style. Okay. So all these things are actually already provided for in your LBY 1 sta and LBY 2 sta. So this is just a um redundancy. However, since it's going to be part of your final exam, I opted to have to allocate screen time on this uh, demonstration, on this review. For the repeated measures design, the main difference, its main difference on the between subjects design is that um, there are 2 points that you have to identify. One is from the perspective of the participant, um, and one is on your own perspective if you were the one who is already organizing the data set or collecting the data. So, okay, from the perspective of the participant, if you were to uh, if you were to imagine yourself being the participant, if you know, if you were um, assigned to at least more than one condition, then in the experimental manipulation, then it means that it's already um, a repeated measures design because um, when you are assigned to when you are exposed to all of the conditions, it means that you have to um you 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 have the researcher should also measure um the dependent variable multiple times because you were exposed to multiple conditions of the experimental manipulation. That's why you also need to take note of the procedure, whether there was a random assignment or counterbalancing that happened. So um, you need to be conscious about how the wording is delivered in the method section on how it played out. You need to imagine the scenario as to what the participants actually experienced during the data collection. So for instance, I wonder why I don't have, okay, this one. So for the within subjects design, notice that there's no group differentiation in that, let's say if we have two conditions, each participant is exposed to all of available experimental manipulation and the measurement of the dependent variable surely will have to be repeated because of the several instances wherein the participant or the test subject is exposed to the experimental condition. So in that case, your data points will be different from between subjects design, um, wherein you can have multiple data points in one case compared to the between subjects design. The advantage in the within subjects design or the repeated measures design is that since you are let's say double dipping on the case, there's already consistency in some of the attributes of the individual. So we call it the individual differences. So this source of variance, or let's just call it error, is now removed from the equation in the within subjects design. That's why statistically we call it to be it is relatively more statistically powerful compared to between subjects design. So if you are concerned with let's say your funds or your time in collecting data, it is advantageous to use within subjects design because it has stronger statistical power. However, it still depends on the nature of the construct that you're trying to capture because it might be theoretically not appropriate to use repeated measures design if there will be a lot of issues with regard to the nature of the construct. Okay, so it still depends on whether it is applicable or not. But if you were to ask me, probably I will use within subjects design by default if I'm trying to test some intervention, and then I will have to determine whether there is whether the intervention is effective or not. So a pre-test post-test is easier than conducting a between subjects design, wherein you need more, at least double the um number of participants in your data collection. So anyway, so it's mentioned here, stronger statistical power, because the individual differences error is removed from the equation. Okay, so uh, another aspect in the within subjects design, so as a specific or a special mention in using paired t-test and repeated measures design is the match design. Match design is somewhat in between. It is neither between subjects nor repeated measures. However, with the way we use the subjects in the analysis, it is referred to as if they were representing one case. For example, if I have two conditions, I will be matching some qualities of those two individuals, let's say by age or by gender, depending on the literature. The literature will guide me with as to what makes sense, what to control. Um, Something that is related to your dependent variable, at least according to the literature. So I will match two cases, two individuals, but in the data points, they're considered as only one case. But in between subjects design, one individual equates to one case. One case refers to one row in your data set. Okay. But for within subjects design, you have one case and you have multiple data points in your dependent variable. But for match participants, um, you have multiple individuals according to your number of um, experimental manipulation, or your levels of measurement, levels of independent variable. Um, you're using multiple individuals, but they represent one case. So they are matched with some particular quality that is related to the dependent variable. However, uh, it is not our it is not our hypothesis. So we are controlling for that. So that's another technique for control of the confounding variable, the match design. Okay, so aside from randomization, standard, making everything else consistent, um, and match design is another technique to do that, to control for uh, potential extraneous factors. Okay, so just a quick look on the how it looks like. So we can just refer here. So the IV and the DV for the between subjects design. Uh, the one column will um, contain your different uh, experimental manipulations, and one column will contain the scores of your dependent variable. That's for the between subjects design. And then for the within subjects design, you have um, you label your first column on your level, on your experimental manipulation or your levels of your IV. So if you have more than two, then you should have, let's say if you have 3 experimental manipulations, then it means that you need to have 3 columns wherein one individual or one case has 3 measurements of the dependent variables. So that's for the within subjects design, and that's what we're going to do. Okay. And then in the match design, so notice the difference here. So in the match design, there are 2 separate, uh, it follows the it follows the data organization in the within subjects design. However, we are um, putting here 2 separate individuals, but notice that they are considered just as one case. So you need to be very sensitive as to how you're going to arrange your data set. So I can test you on that from time to time, test your understanding. Again, according to cognitive psych research, you need to test yourself. You quiz yourself. Okay. So just a continuation um on the use of within subjects design. The caveat of using repeated measures design is that we need to be cautious with our internal validity, or we might be introducing some confounding factors that um, that predicts the variance of your dependent variable. For instance, we have what we call the order effect. So the order effect, regardless of um, your conditions, if you have consistent um, sequence on the delivery of your conditions, we don't know what exposure of one level can have on the next level. So in order for us, if there is such an error, we do not want it to systematic. So we break it apart, and we want to make sure that the source of error is evenly distributed theoretically. That's why randomization can also be used in the repeated measures, or counterbalancing can also work. If you do not have a software that will allow you to randomize, at least counterbalancing, a manual way to do it or an analog way to do it is to plan how they're ordered. Right? So for example, you can experience fatigue um in, let's say, the tasks that you uh, are doing when you participate. That's why in the latter part of your performance, it might get depleted because you're already tired. So assuming that you have the same sequence of uh, exposure of the conditions, then your interpretation might already be fallible from the order effect and the fatigue effect. That's why counterbalancing and randomization, and if not, counterbalancing is also important in order for us to um, rule out that kind of explanation of that effect. Because if you cannot rule that out, we're not confident of our interpretation anymore. Then your data will be useless. Your study will be useless if there is an implanted doubt that you cannot resolve. Okay. So that kind of doubt already make your work like a trash because you cannot make sensible conclusion out of it. What's the point of conducting an experiment if the design is not rigorous and the interpretation um is fallible with all of the systematic error that can come um in accounting for your patterns in your dependent variable. Okay, so pre-test and post-test can also work as well in longitudinal design. For example, in developmental studies, um, okay, for example, in developmental studies, you're not really providing an intervention. However, um, the time in itself is already working on its own pace. So that is a different that won't will not be considered an experimental study because you as a researcher did not provide any intervention at all. Okay, so longitudinal design is different from experiment. So you want to make sure that the changes is because of your um experimental manipulation and not because of the changes in time. Let's say in a longitudinal design wherein the developmental aspect, it is already given that something will change eventually. Okay, so let's proceed. Okay, so what are the uses of paired samples t-test if you want to compare two means from the same individual? Okay, just like in pre-test, post-test, or two different but related conditions. So in behavioral genetics, for instance, the pair of twins are considered one case. Okay? So that's referring to match design. So you can use paired t-test in that. And then uh, again, the goal is to test the mean difference. So it can be it will depend on your goal. So for instance, you want to test the difference between two time points, or let's say for longitudinal, or difference between two time points wherein you have an intervention in between, or difference between two conditions. Let's say you have two experimental conditions, uh, or you have two measurements at least, and then a match pair. Okay, so um, this is just the hypothesis test um, statement, and I'm sure that you're already familiar with this. Okay, so let's try to continue with our lecture. I think this is already exhaustive. This is just a review anyway. Okay. There are also some concepts that are applied in repeated measures design. So it somehow it's a bit more complicated than between subjects design. Between subjects design is simpler because you don't have to think about how you're going to arrange your stimuli. You simply have to um, prepare them, and that's it. You do not have to um, put more extra effort on that. But in repeated measures design, because we are concerned with the order effect, we need to plan a lot in designing the task. So for instance, in this example, we have multiple trials. When you say multiple trials, you have set of items that are uh, that that con- that contains um, multiple stimuli, but you are manipulating the independent variable accordingly. So for example, my um, manipulation has something to do with different facial expressions. So I may need to use 25 trials because if I only use one trial, then it might not be um, very informative because I'm using aggregate score here. Okay, so refer here for instance, I'm asking a question about um detecting the most preferred, okay, so the most preferred most liked facial expression. So let's say you have to rate rate uh how much you like each face, then you need to present them and then gather data for each stimulus that you present. So I remember having had students who manipulated who conducted repeated measures design in their final paper wherein they manipulated the width of the forehead, I think. I can't remember what they manipulated, but it has something to do with morphing some facial feature. Okay. So for instance, I have this block and so on and so on. So this is trial one. Okay. And then on the next trial, there will be a different set of uh, faces. So let's say in this trial, maybe I'm looking at, okay, maybe the manipulation here could be the smile. So how open is the smile? So let's say my manipulation here is um wide smile, wide uh, nose, open wide. Okay. So the actually the manipulation here is when they appeared again on other trials. But with if I can morph, for instance, I have a software wherein I can morph their smile, wherein the first this same stimulus will have different versions wherein it will have a closed smiling version and open narrow version. So it will reappear on other trials. Um, and then that's why it has to have multiple trials because um, y- the researcher need to determine whether you are consistent as well in your response. So it depends on the design. So for instance, I have another set. I'll create another set. Maybe I will look for male. This is just male. In counterbalancing, if your manipulation is this one, the open, narrow, closed, and open, wide, so you need to have a different sequence on the next one. So let's say this is B, B, A, C. So for this one, this is A, B. I need to look for another one. A big gate, a big, a wide open smile for male, for example. And okay. Okay. So in this case, this is closed, open, narrow, and wide. It requires a lot of preparation in and a lot of thought in within subjects design actually. If we are using uh block randomization. So in this case, what I'm doing now, if I plan this kind of combination, then this is called counterbalancing. So the block randomization can come if, let's say, I have a software wherein I can prepare one block. So let's say I have a set of about 5 or maybe 10 trials. Okay, so let's say I have so 10 trials per block. Okay. So then I can do another one, another set wherein they will reappear, but they have different conditions. For instance, uh this female lady will appear on the next block as um open, narrow. I can't edit it. Why is it? Oh, so block 2. I can't edit it. Okay. Mm. So I will combine it with other, for example, it's ABC. Well, it can be the same, but the combination will be different. The morphing the morphing will be different. For example, if I have a way to morph this, uh, can I draw here? Well, I can't draw anything here. So I will find a way, for example, to morphed version, open, narrow version. Okay. So that's how block randomization works. So it's more complicated than between subjects design. That's why it will require a lot of thoughts. It will require a lot of preparation to in the materials for repeated measures design with this kind of experiment. Okay. So counterbalancing, you already know that. So all I need to do is just to plan the sequence of the different permutations of your combined permutations of your uh, experimental manipulations. But in block randomization, it is more complex, like in this example. Okay, so just to properly define it, it's a method of randomly assigning participants that guarantees that the condition sample sizes are equal or almost equal. So what we're doing here is we're assigning the set of participants into set of conditions and then assign them to another conditions. So that's just how it works. Okay. So let's continue. So the principle is actually the same. We're just trying to remove any potential source of systematic error. So it's the same goal anyway. Okay. We already mentioned this. So a complete design is if you administered the full combination or full permutation of any combo that you can get from the number of levels of the IV that you have. Okay. So if it's too many, then it can be just partial. Then you can use incomplete design. Okay, so let's just continue. So I want you to do this. Can you prepare your statistical software on your laptop? Okay, so let's try it first. What are you using? Are you using Jamovie or just? So after this, I will be giving you a data set, a file data set in this. Okay, so I want you to open that and encode or import the data set in however way that you can. Okay. Can we begin? Can you raise your hand if you're already prepared your Excel sheet and your statistical software? Raise your hand. Hello? Okay, Kenji is prepared already. Okay. So I have an example here, an actual data set from from my master's thesis. So, but I will just show you the variables that you will need. Do we need to input the data in both Excel and Jamo? Well, whichever case that is more efficient for you. If it is more efficient for you to import the Excel sheet on the Jamovie or JASP, then do it that way. Or if you are comfortable encoding it straight to Jamovie, then that's fine as well. So again, what's the routine that we do here? Identify the independent variable. So the independent variable here is time. So before and after. The dependent variable here is the weight. Okay. Yeah, sorry. The independent variable is whether you actually attended the program. So it's before attending the program or after attending the program. But in your data set, it can be put just as before or after. So I want you to conduct the analysis now. So you just go to your Jamovi or JAS. So go to analysis and click paired samples t-test. Okay. And then input your data set side by side, and then you can also practice taking the normality checks. And when you do that, I also want you to take the effect size. Okay. So for the normality check, usually we're happy that Shapiro-Wilk is not statistically significant. It says that it is normal. And what else? Okay. So this is just a descriptive stat and the descriptive plot. You can determine which is higher or lower if you see your descriptive stat and plot. But definitely what will help you determine if there is a statistically significant difference is when there is can I annotate here? So this one, wait, oops, the p-value. So if the p-value is less than 0.05, then it means that it is there is a mean difference, and then you can tick the plot so that you can see the difference visually. Okay. So how are you going to write it in APA style? So if you're done, I want you to paste your work here. Later, I will be giving you a data set, and then um, I want you to repeat the same. Were you able to follow the procedure here? It should show this. Okay. So now I will be providing you a real data set from my MS thesis. But only the relevant parts. Or you can paste the results. Viewer. Sorry. Ayan na. Naka-editor na siya. Don't paste it yet because ito pa lang yung i-require ko. That's just an first exercise. So now this is the graded exercise. I'm just looking for the file. Okay, there you go. So there it is. Can you see my chat? Okay, so download that, and then I want you to conduct the same analysis pre and post affect. So in that data, I measured their affect. So with using a Likert scale of point, I think it's 0.05 to -5 to 5. I cannot recall exactly. I think it's 1 to 7. Um, I will have to review it. But anyway, it's just a measure of affect wherein the higher it is, it's using a continuum wherein it's between negative affect and positive affect. So the lower it is, um, the 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 higher it is, the higher the positive affect. So that's how that particular scale is used. So ang dataset nito, hindi ko na-explain yung context, pero ang conditions na lang na ipapakita ko sa inyo dito is merong conditions na nakalagay na un- understated, accurate, and overstated. So these are the three different conditions that are used. So how do we apply that in the repeated measures analysis? So I'm going to demonstrate it to you, and I want you to paste it here as well. Third test and pay. So I will demonstrate how to use repeated measures ANOVA. Okay. So notice here, so it shows the data structure that I've been telling you about in the past few minutes, wherein I referred to one case having multiple measurements of the dependent variable. And the name of the columns here refer to the experimental manipulations or the level of the IV in your repeated measures ANOVA. So I will click ANOVA and then click repeated measures ANOVA and then relabel this, let's say, visual feedback. So I will just put here what are the names of these manipulations. And then all I need to do is just to put it there, match it according to how it appeared on this part. And then voila, you already have your repeated measures ANOVA results. And I will take eta squared and post hoc test. And you can also put the descriptive plots. So here's the result of my repeated measures ANOVA. Okay. So how are you going to interpret this? So if you see here your repeated measures predictor, you will find the p-value to be less than 0.05. And then it only tells you that at least one condition is different from the other conditions. And in the post hoc, you can tell how is it different from each other. So this condition is 0.07 higher than the accurate condition. And it is according here, so it works as a p-value. So according to this post hoc test, this condition is statistically different from this condition, and so on and so forth. So it just looks so clean, but it can have different scenarios in the ANOVA. Okay, so I will now wait for your output. So kindly follow this. So if I made a paired t-test here, then I want you to immediately follow a repeated measures ANOVA.